Welcome back, fellow mitochondriacs, to another episode of Cancer is a Mitochondrial Metabolic Disease. I'm going to give a quick introduction into part two with Mark Lintern of the cell suppression theory. In this part, I had some basically doubts about some of the likelihood of people having these fungal infections based off of my own personal clinical experience. We talk about, could a ketogenic diet be harmful if the cell suppression theory is based on the cell danger response theory? We talk about some of the contentions that Mark has with the mitochondrial metabolic theory. Is there a reason why some of these repurposed drugs like ivermectin and bendazole, statins, metformin, etc., have anti-cancer effects apart from their effects on the metabolic pathways? We talk about mitochondrial heteroplasmy and the bioenergetic decline that is proposed by several research groups as the underlying cause of the metabolic theory of cancer. We talk about hypoxia, hypoxia-inducible factors, and some of the factors that lead to pseudohypoxia. We also talk about salvesterols. We get into cancer stem cells and much, much more. So if you liked part one, I have a feeling you're going to like part two. So let's get into it. For me, in some ways, and I'm maybe a little biased, maybe more than a little, um, I think it's hard for me to wrap my head around the idea of having such an occult infection that that causes this process to happen, activates the cell death, cell danger responses and has all this inflammation associated with it. Yet, there's really no symptoms from a patient, I think it's hard for me to wrap my head around, I guess, because, if you other, intracellular bacterias, you'll have the symptoms of a classical infection, fever, chills, body aches, malaise,
Yeah.
and maybe other things, right? Like I think that if I'm not mistaken, I read is it Straussman? Is that the guy? I think he
Yeah.
talked about some of the common, fungi that are potentially in these tumors, and they're candidal species, whatever. Candida causes thrush. Candida causes Candida esophagitis. There's some kind of like sign somehow, that that person has an infection. And, the classically inflammation is like the rubor, calor, whatever, like the redness, warmth, et cetera, that happens with the inflammatory response. So it just seems like for me a little bit hard to wrap my head around. And on top of that, like I'm not saying you're wrong or
No,
Straussman's
no,
wrong,
no, absolutely.
nothing
I've
like
got an
that. I'm just
answer
saying like I have my
to
own bias, and I've
sure.
had the chance, throughout my years as being being in the hospital and of culturing every piece of a body, like I mean abscesses everywhere, you know, pretty much blood, urine, stool, you name it, and there just is not a lot of, and I'm not saying those cultures are able to capture always what we're talking about here, or maybe never. Because it seems like I never really see like significant like fungemia, most patients, or like or fungal infections in culture. So I guess it's not that I'm necessarily, apart from being, skeptical but still open-minded because I want to help people, you know, what is the clinician to do, to find these infections? If the patient is asymptomatic, there's no real way to conventionally, obtain a sample of these fungi, know whether or not you need to treat it, what you're treating, what kind of clinical response that you're having, classically how we treat, practice medicine. What is one to do with that information?
Well, I think firstly, the issue is that no one's considering that fungi, as I'm saying, are the underlying mechanism driving the disease. So
Fair point.
you're not specifically looking for it. So you're not going to find what you're not looking for. The other thing is it's a novel opportunistic infection. So what we're finding now um, seems to be the case is that most of the tissue in the body has its own microbiome. So you'll have a homeostatic microbiome where you don't have a dominance or dysbiosis in any particular pathogens. So you have homeostasis within the tissue. But what I'm saying is the reason for cancer forming, it's not that you'll have an overt infection because the body will deal with that. What you have is the slow progression of biofilms being generated in particular tissues which harbor particular pathogens, that when the moment of opportunity arises that the environment benefits them to then transform from their yeast form into their more invasive hyphal form, they will take advantage of that. And that is where chronic inflammation comes in. So you have chronic inflammation in a particular tissue, then you have iron overload. Iron is rocket fuel to pathogens. You have lactic acid being produced because, this is the repair process going on in a chronically inflamed tissue. So you have overproduction of lactic acid. Both iron and lactic acid suppress immune cells at these sites of damage, but they both fuel and feed the pathogen. So now you've got an environment in a chronically inflamed tissue, say, where you might have a microbiome that is homeostatic to a certain degree. You've got the addition of antibiotics in food and or overprescribed to patients. And now you're getting a slightly dysbiotic process where you've got the potential for dominance with a certain particular fungal pathogens. And in that process of that inflamed environment, you move into a threshold where uh, a small intracellular infection can occur within that tissue. That occurs, and it's not a case of you have external infection ravaging the tissue. It's that you've got this intracellular infection that has happened. Inflammation makes it easier for pathogens to invade cells. And if they manage to establish themselves within that chronically inflamed tissue where the immune cells aren't functioning as well as they could be because it's prolonged inflammation, prolonged lactic acid, prolonged iron overload, you have a situation where the pathogen can sustain itself. And if it can sustain itself, it can then start modulating the local environment. So one of the things that the pathogens do is trigger a phenotype from an M1 to an M2 macrophage phenotype and a Th1 to a Th2 phenotype in the local environment. So it's not that you're searching for and you should see mass infection, mass fungal infection throughout the body. It's that the local tissue in that particular area, for whatever reason, whatever carcinogen is causing that damage in that particular area for a prolonged period of time, that has enabled this opportunistic infection to take place. And then the immune system is having difficulty actually getting inside to eliminate that.
And it sounds like it
And
has to be a pretty benign, not in the way of cancer, but I mean a benign process in some ways because it can't be an overwhelming infection, right? It has to be like a
no,
low
of course.
level, just
Yes.
there, kind of
Yes.
irritating the tissue, kind of a thing,
Yes. So
causing, yeah,
sorry, what I was going to say, what we see is like with Candida, that they sit within the biofilm and they produce acetaldehyde, one of many metabolites. And that metabolite, actually, what you see when that increases, you see a reduction in um, oxphos capacity within the mitochondria. So it's almost like they're terraforming the local environment to force this switch over to the Warburg effect. And it's prolonged use of the Warburg effect um, actually facilitates infection. That's the problem. That's why we have why chronic inflammation, I say, is associated with cancer, because it benefits the pathogen. And what you'll have is this going on all the time. And because most of our organs are colonized, it depends on your diet, it depends on how much inflammation, how exhausted the immune system is at any given moment in time, and what the colonization status is of any given tissue. And if you have a dominance in particular pathogens because you're overweight and you're consuming too much glucose, which again is feeding the pathogens within the body, you have this danger of this successful invasion intracellularly of a number of cells. And if the immune system cannot eliminate that, you then have this cascade of events where the cells around it also trigger the Warburg effect, and they're caught between CDR1 and CDR2 in Robert Navio's cell danger response model. That's what I propose there is going
got
on.
it. But I mean, just let's think through that just for 1 second, because it
Sure.
seems like in some ways it would be very problematic. Um, Because if the pathogens, are glucose dependent, like we're talking
Hmm.
about, then upregulating glucose transporters and glucose utilization seems like that would be a wrong strategy. I mean, I'm not trying to question God here, but I'm just saying that like it just seems like it would be a poor strategy, number one.
Well, not
Um,
for the pathogen if it's accessing the glucose that is entering the cell.
if it's accessing the glucose that's entering the cell, but it would be available to the intracellular pathogen, though, right?
Yeah.
Correct. So what I'm saying is, it seems like although that is the response that Navios found,
Right.
cell danger
Sure.
response, it seems like that would be in some ways beneficial to the pathogen because it's giving it a fuel that it would need, right? And then I think my understanding is that fungi can also use glutamine as well, right? So HIF1,
And fat.
not only does it upregulate glucose utilization, but glutamine utilization. So it seems like it's almost like we're feeding the enemy in that case, if we're calling the
Yeah,
fungi the enemy. And
exactly.
then, you know, um, to be fair, going upon that line of reasoning, it would seem that if the normal response to healing is a increased glycolysis in phase one, aerobic glycolysis, Warburg metabolism in phase two, it almost seems that a ketogenic diet at that point would actually be harming that, because you're limiting the amount of fuel it needs for the regeneration and the repair of tissues. And I think that that's where maybe there could be some at odds with the, therapeutic modality of Seyfried's metabolic theory of being on a ketogenic diet, knowing that the normal cells are metabolically flexible and the cancer cells are not, by his theory. Um,
So,
but what we're saying is that the infected tissue is transiently, as a suppression theory, like you mentioned, being transitioned for a period of time of healing. And if we were to limit that, then potentially we could harm ourselves in some way of looking at it, right?
well, I suppose the nuance is this, that an acute infection or acute response actually is beneficial because it's able to reduce the amount of glucose that is available in the environment, because we're looking at an infection. So the cell won't be alerted once the pathogen is present within the cell. You've got this process of the pathogen invading the cell. So at that time, you've got this acute infection occurring. Glycolysis switches over. It alerts the immune system into action. The whole process of this stifles the ability of the fungal pathogen to invade the cell during that whole entire process. And hopefully, you've got help from the immune system coming in, neutrophils invading intracellularly to eliminate the toxins being produced and the pathogen
Right.
itself. And then everything's resolved. So the whole process is really effective in the acute phase.
Right.
But the problem occurs when it's chronic. The chronic inflammation reduces the capacity of the immune cells to operate. You have immune exhaustion as well. It can't access the cells. And once the cell is invaded, then it becomes beneficial in the sense that you're now driving glutamine and glucose into the cell. It makes, because what the pathogen does, it sequesters the nutrients in the cell. It sequesters the pyrimidines, the purines that it needs for its own
Iron,
proliferation.
etc. Right.
Iron, arginine, methionine, and so what you get is this process where the pathogen is actually absorbing these nutrients and the cell, because the pathogen suppresses apoptosis, and this is one of the other key features, it needs to survive. And apoptosis is a defense mechanism, is a last ditch defense mechanism for the cell. Hit the self-destruct button, eliminate the pathogen in the process. But the pathogen suppresses that process, upregulating BCL2, blocks the mitochondrial cell death process. And because that happens, what you now have is you have a pathogen in control of the cell, absorbing or obtaining, sequestering all these nutrients, and the cell is now working on autopilot. So it's always trying to establish homeostasis. So it's now depleted in pyrimidines, it's now depleted in arginine, it's now depleted in methionine, it's now depleted in glucose or glutamine. So it upregulates those processes because the rest of the cell, it's not like the pathogen blocks everything in the cell. It needs certain specific functions of the cell in order for it to survive fully. So it fully understands that it needs to block the defense mechanisms whilst upregulating other things so that it gets access to these nutrients. So effectively, in the chronic phase, when the pathogen has established itself intracellularly, it's actually the Warburg effect is a beneficial effect. So then when you're talking about, so now we're talking about cancer effectively. So when you're talking about a ketogenic approach or fasting, what you're going to be doing is you're then reducing the primary fuel of the pathogen by blocking the glucose initially. I mean, the pathogen's cell wall is predominantly carbohydrate, so that's one of the main reasons why it requires so much glucose. But that's why a ketogenic diet will be beneficial because it's blocking now. It's now the actual cancer cell, which is the vehicle supplying the fungal pathogen, that
Got
is
it.
the issue.
I still think though that it somewhat muddies the waters because whether or not the cell is quote unquote terminally damaged or not, it is
Yeah.
now effectively commandeered by the enemy. And the only way to really win, I guess, the game, I guess you'd call it, the host winning, would be to kill that cell off by apoptosis, and treating it like the enemy in some ways, because otherwise you're just allowing the
Mm,
chronic infection to continue.
you could do that, but then you've got the process with the problem with chemotherapy, haven't you? If you're using toxic cytotoxic compounds, the body and stem cells are going to resist that and upregulate efflux pumps to prevent that. You're going to get resistance. What I see in the medical literature, or what I feel, is that working with the body is far better. So you want to be stimulating mitochondria with hyperbaric oxygen therapy. You want to be able to methylene blue. You want to be stimulating mitochondria to actually continue to do the function they're trying to do, which is use oxygen to eliminate the pathogen. They are the defense mechanism in the cell. And then by using a ketogenic diet or fasting, you're restricting access to glucose for the pathogen on a number of different fronts. You're inhibiting the pathogen's ability to utilize that cell for its own ends, and you're increasing the defensive mechanisms against them. So you also want to obviously switch the Th2 response to a Th1 response. So now you've got, rather than cell repair um, uh, immune cells surrounding the tissue, absorbing arginine and various other compounds and glutamine, you want to stimulate those intracellular immune cells to um, get into the cell more frequently to eliminate the pathogen. And this speaks to something you also mentioned. I was listening to your talk with Thomas Girai or Giraj.
and
Not sure how you pronounce
it's
his.
Jirai, and I always say
Right.
Diraj, and I feel bad because he told
Yeah,
me.
Girai. Okay, great. Um, and you posited a question which I thought was really interesting, and it was about the flavonoids and polyphenols in
Hmm.
certain natural compounds, specific berries. That there's a paradox there because the berries are high in glucose.
Glucose, correct.
And yet there's still an anti-cancer effect. So technically, the berry should be um, feeding and fueling the cancer. So what is the mechanism there? So one of the things I point out is you also have a similar thing with honey. A number of studies show that if you administer honey to cancer cells and healthy cells, cancer cells trigger apoptosis, the programmed cell death mechanism that is suggested to be defective because mitochondria are defective. Yet that's reinstigated, which suggested to me there's a suppressive mechanism going on for one. And two, how is it that honey, primarily uh, sugar-based, glucose and fructose, how is that not feeding the cancer cell? That should be feeding the cancer cell. What is the other feature of that? And similar along those lines, you've got silver. So if you administer silver nanoparticles, um, in those studies, we find that healthy cells are not eliminated. They're perfectly fine, but the cancer cells trigger apoptosis. Again, that suggested to me this whole point of this process of apoptosis isn't defective because mitochondria are defective. It's suppressed because the pathogen's
Yeah.
suppressing it.
I don't
And the
know. I don't
reason,
know, Mark, like where and I mean the literature is vast, so I don't know what
sure,
author is saying this, but what I'm saying is I have not seen anybody really suggest that apoptosis is fundamentally broken. I do believe that it's well documented that it's suppressed. And I think it's suppressed by two major mechanisms. And it's both controlled by HIF1 alpha, believe it or not. Again, HIF1 alpha will upregulate an enzyme called pyruvate dehydrogenase kinase, PDK. PDK effectively shuts down PDH or PDC, which is pyruvate dehydrogenase complex. That is what converts pyruvate, the end product of glucose, into acetyl CoA. And it allows it to not get into the TCA cycle to function. And it kind of forces pyruvate into lactate. Okay. So that's one mechanism. The other mechanism is that HIF1 will directly shut down OXPHOS at the level of the actual protein complexes as well. And in that it also shuts down intra-cancer cell mitochondrial melatonin production, which I think is also very protective for the cancer itself because melatonin has got profound anti-cancer effects. So my point is that it's shutting off the switch. HIF1 is shutting off the switch. The reason why I believe that what you're talking about is possibly restored is because there are several drugs, supplements, etc., that can reverse that process, like you're mentioning. I could name you probably 5 or 10 right now. DCA is the prototype here, okay? Dichloroacetate. But it blocks PDK, so therefore it's uninhibited. pyruvate can get into the mitochondria, and I believe it's the addition of energy to the system through the normal channels. It's what leads to increased reactive oxygen species at the level of the electron transport chain, which then allows for the cytochrome C, which is the trigger of the intrinsic apoptosis, to be allowed to be ejected from the mitochondria, and you are able to restore. Same thing happens for curcumin. Same thing happens for resveratrol. Same thing happens for thymine. Same thing happens for quercetin. Same thing happens
Mhm.
for ivermectin. That's also possibly one of the mechanisms of how metformin works, because it also causes complex 1 to be leaky of reactive oxygen species, more ROS, peroxidation of the lipids, and then apoptosis. So I agree fully, okay, that apoptosis is likely suppressed. But I do think that that's explained by available science, and I don't think it's, one of the things I've mentioned, I'm not trying to be like adversarial here, but like I've heard you say there's three things, at least three things that you think that is missing from the metabolic theory, one of which is the lack of control of apoptosis. The last ones that you had were immune evasion and also the lack of consistency to explain the mitochondrial function issue. I don't know if those are still true for you. It seems like at least you've come to terms at least that lactate is responsible for at least partially suppressing the immune system. So
Hmm.
maybe there's only two things now. But yeah, I think the apoptosis thing, I think you're right. It's just that I don't think it's at odds with, with what Seyfried's saying.
Sure. No. Well, I mean, this is the point, isn't it? There's that explanation you've got there, but there's also the explanation that I've got as well, and both seem to be equally um, possible. Um, the point was going to make was that a lot of those polyphenols and flavonoids, honey and silver, are antifungal.
Hmm.
So I would argue that the process being undertaken here is an antifungal effect within the cell. You mentioned ivermectin. When you look at that's also antifungal. So is fenbendazole, mebendazole. Most of the off-label drugs, even lovastatin, statins that are used, metformin. When I've looked at all these as well to try and find commonality between everything, the consistency, that's what I've always tried to find. The consistency is that all these drugs are also antifungal. So I'm arguing, okay, so we're also targeting the metabolism of the cells, yes, but are we inadvertently not realizing that we're actually targeting the fungal pathogen through this antifungal mechanism of these off-label drugs, of these natural food compounds? Because I note that sulforaphane in broccoli is highly anti-cancerous. And I was looking at some of the work Dr. Michael Greger was talking about, and he
Hmm.
in his book he discusses how they took tissue from breast cancer patients, but before they did that, they gave them broccoli as a dietary
Hmm.
implement just to see if the sulforaphane would transfer and be present within the removed tissue, the cancerous tissue. And they found that it was present. And we know that sulforaphane in that form is antifungal as well. And then if I don't know if you've heard about salvestrols and the work of Professor Jerry Potter and Dan Burke from um, De Montfort University in the UK.
I don't think so. I've heard of sylvestrols, but I don't know what they are or what they do.
So they were working on CYP1B1, an enzyme which is upregulated in cancer cells. But to utilize that enzyme and use prodrugs to try and develop those drugs so they would respond just in those cancer cells by being broken down or metabolized by that enzyme. And what they found when they were looking at this, Jerry Potter discovered that the molecular structure of the prodrugs they were trying to create mimicked similar molecular structure of some of the polyphenols,
Hmm.
the natural compounds found in plants. So he did a search to see if he could find any particular compounds, which he's now named salvestrols. And he found that what happens is when those salvestrols, such as resveratrols, one from
Hmm.
red grapes, highly anti-cancerous and highly antifungal, um, they're absorbed into the cell and they're broken down or they're metabolized by that enzyme into a compound that is highly anti-cancerous and highly antifungal. So he's arguing that there's a mechanism through these plant compounds, as long as you're utilizing organic plants which produce greater polyphenols, that because those polyphenols are a defense mechanism against pathogens in the field,
Hmm.
then the body is utilizing, the cells are able to utilize those antimicrobial compounds to effectively weaken the pathogen as well as everything else the body is doing to try and eliminate the pathogen, which is why food can be seen as medicinal in that sense. Um, but also silver, I mean, the big thing is why would silver trigger apoptosis in cells? Uh, Maybe there's some connection, as you're saying, with HIF1 alpha, possibly being downregulated. I don't know, but I'd have to look into
I have
that.
to study it
But
too. I'm not sure the mechanism there.
no, but
I'm sure there's an explanation for it though.
well, the mechanism for me would be that silver is highly antimicrobial.
Hmm.
That's why they use it in the space
Oh yeah,
station line.
I know.
You know,
Integrative
so
medicine, they've used that for a long time. What I'll say, what I think, what I would challenge you, Mark,
sure, please
is
do.
that, and I have to challenge myself because I also write things off, a lot of times because I'm looking through something through a very certain lens. I'm like, okay, does this compound have effects at the level of glucose or glutamine, right? And what I'll tell you is that everything you mentioned has pleiotropic effects. It is
Mm,
basically curcumin is the
sure.
poster child, right? Curcumin probably has like 30 or 40 different mechanisms, right? How can we say that it's the antifungal effect or it's the PDK effect or the inflammation effect or the glucose or glutamine effect? It's almost probably impossible to know. It's the fact that it has so many diverse mechanisms that it's so powerful. And I don't know of all the fungal effects of all those things. I know things like garlic and berberine and olive leaf and things like that. I mean, because we use them in functional medicine to treat like pathogens in the gut, but I don't know of all those things. And I don't know that I don't personally know of an antifungal effect of metformin or resveratrol. I don't know those. I mean, I'm not saying you're wrong. You actually are right, probably. I don't know the answer to that. What I'll say is that, and this is kind of what I would, if you were, uh, and I don't want this to come off as paternalistic or as downplaying you at all, okay? But I just
Sure.
have to relate it to my own life, right? So I teach medical students at my that are residents, excuse me. And what I would tell them is to expand your differential diagnosis, right? Like if someone comes in with chest pain, it's not always a heart attack, right? It could be grandma fell and fractured a rib and she has a pulmonary contusion and or she could have a pulmonary embolus or a pleural effusion or a pneumonia. There's just a lot of different things that it can explain a very similar syndrome. And so my point is that I'm going to try to challenge myself to include fungal infection and that mechanism for certain polyphenols and flavonoids and drugs. But I would also ask you to just say that, like, I think we would be just as bad as the researchers that we don't like, saying that the reason why, metformin helps, is this because it does this one thing, where it maybe has
Mm,
diverse effects, and just
sure.
realize that there's no one's really going to probably ever know what the real reason is why curcumin at 8 grams, doubles life expectancy for pancreatic cancer. There's probably a dozen reasons why, one of which could be 100%. I'll concede that it could be a chronic fungal infection that it's
Mm,
targeting. And that's the main effect. It could be that the reason, but
sure.
I think it's, it's just, what the reason why I tell you this, man, is because, um, when you are so sure, and I'm not saying you, I'm saying like when my clinicians that I'm teaching
Yeah.
are so sure that the reason why grandma's chest pain is she's having acute MI and you didn't ask her, did you fall? Do you have tenderness here? You missed the rib fracture, you missed the pulmonary
Mm,
embolus. And I just don't, I want to make sure your theory is as complete as possible. It explains all the freaking, hallmarks and more. And you win a Nobel Prize, man, because I don't care about glory, man. I want people to be well. And I'm sure that's the same for you. And I just think that in some ways, iron sharpens iron, And so I think that if it's okay with you, can you still please explain to me? Let's say we've handled the apoptosis issue. Do you still have the other three contentions or the other two contentions with the metabolic theory as being incomplete?
Uh, well, the one major contention would be I'm questioning whether mitochondria
Are damaged?
are damaged to the extent that OXPHOS is dysfunctional, and then that is the underlying mechanism that drives cancer. Um, With the immune system issue, I agree. As you mentioned about me mentioning lactic acid being suppressive. Um, yeah, I agree with Thomas Seyfried that that explains, and another other things he points out explains that issue later on in the disease. But the issue I've got is what causes the immune system to ignore and not see cancer cells at the initiation stage when the lactic acid isn't at the point where the immune cells can't identify um, that that stage.
I
So
think
for
that's a
me,
million dollar question.
that doesn't explain it in the early stages. So I'm still, yeah, that's still a
I
contention.
think that's an excellent question. I think the initiation stages and the etiology of cancer or the cause or causes, I think is what where the gray area is. And I personally believe that it's going to be everyone has a very I personally believe everybody has their own n equals one experience of how you ended up there, because, I'm not a psychologist or psychiatrist, but I'll tell you that if I had a nickel for every time someone told me they had a terrible thing happen to them, 6 months or a year before their cancer diagnosis, and I'm talking terrible things, like traumatic things, I would say that it's hard for me to think that that isn't contributing somehow, you know, I don't know exactly
Yeah,
know how, but it
well,
is,
well, I do processes. I explain this in my book. So what I've tried to do with everything in my book is draw everything down to consistency. So one of the extra things I wanted to explain was why so many different carcinogens can cause cancer to develop.
Mhm.
What's the consistency there? Why are nurses, night nurses, got a higher risk of developing cancer because their sleep patterns are, all over the place? Why does stress, like you say, people tend to recall an experience, a negative experience, a life experience before their cancer development. And when you look at this process from trying to explain it in a consistent basis, it's because all those processes facilitate infection. Even the mutations that are said to drive cancer, the primary mutations, they facilitate infection. So what in that process you have chronic inflammation, you have exhaustion of the immune system, you have the generation of this dysbiotic microbiome, um, and emotion, the driver of cortisol. You're creating this inflammatory environment which actually benefits the one thing, which is the pathogen, and suppresses the immune system. And that's the same with any other particular um, carcinogen, because what you're doing is you're generating an environment where you've got ion overload, which again fuels the infection. You've got the chronic inflammation developing, fuels the infection, suppresses the immune system. So you're creating this environment. Doesn't matter what type of carcinogen it is. Any carcinogen creates immune suppression, and all these factors that then lead into facilitating this process of infection. And that's the core consistency that you can see occurs. Doesn't matter what the multifactorial underlying driver of the condition is,
Mhm.
it all comes down to the certain key factors, and those key factors, even mutations, like I say, they drive this infectious process. They increase the ability of fungi to invade the cell and gain control.
Yeah. I think that it's funny, man, because I mean, obviously you're convicted. And
Well, like I say, I'm open to being wrong,
no,
and
but
like you
I
say,
sense your conviction, and I actually I honor it and respect it. It's just that I feel, the same way about all of those things leading to progressive mitochondrial damage,
hmm,
and I don't think that we're going to find a necessarily common ground there, which is fine. I'm okay with that. I guess what I would tell you is that there's a researcher named Douglas Wallace. He's at the Children's Hospital of Philadelphia. He coined a term called mitochondrial heteroplasmy. I learned about it through Jack Cruz, who's a doctor I follow, who's brilliant,
yeah, I
but
follow both of those too.
he's hard to kind of follow sometimes because he's so
But it's interesting his interpretation of light
he
causing
is,
cancer.
oh my God, dude, he is brilliant. I love him,
Yeah.
but he's hard sometimes to swallow. Sometimes his pills that he drops. But my point is that through him, through Wallace, and through other researchers that are in kind of the same fields of mitochondrial genetics, et cetera, the consistency is the bioenergetic decline is what they talk about. because every mitochondria has their own mitochondrial DNA. Every circular plasmid DNA has 37 genes and codes for OXPHOS proteins only, right? So you don't have to have like a terminal mutation like you mentioned like Barth syndrome. You don't have to have a terminal mutation. You have to just hit it enough and have a damage to where the electron transport chain protein is leaky of electrons, and that will snowball into creating more reactive oxygen species, lipid peroxidation, lack of crystal formation, lack of supercomplex formation, and bioenergetic decline. And if you look at the graphics from Seyfried and from Wallace, who don't know each other, I confronted Seyfried about this. I'm like, do you know Wallace? He's like, no, I know of him. I met him. I think I maybe met him once at a conference, but I don't know him. So they're completely different areas of science. It's not like they're collaborating and they're like, sharing notes. They're multiple areas of science, and they come to the same conclusion that it's the gradual bioenergetic decline. And at some point, you hit a threshold, just like Seyfried has a T in the middle of his little graphic. I think he means
Yep.
transformation. But my point is it's very similar. And then another group took that and they looked at what the metabolism looked like of those mutant mitochondria, of the mutant DNA. And it has upregulation of glucose and glutamine. And it's just like it's hard for me to reckon that, as being too coincidental, and then, I don't know. I think that's what I would say is that I think that's hard to kind of conceptualize that it could be anywhere on that nuclear or that mitochondrial DNA as being damaged, but it's extremely susceptible. So, because it's right next to the ECT, the electron transport chain, where there's a lot of ROS coming off. There's not as
Sure.
much protection for it, and it doesn't have to be consistent of this always this gene, because ultimately any gene on that genome will affect mitochondrial performance because all those genes encode for is OXPHOS proteins. So I just wanted to put that out
Yeah,
there.
I'm not sure if it's mentioned in Sufi's latest paper, but he actually points out that when he tried to identify pathogenic mitochondrial DNA mutations that would explain
Mhm.
OXPHOS deficiency, he couldn't find it, and that it's controversial.
It is.
So that's
It is. I thought
so,
he told me that. And but then
and that's one of the
but
reasons why I'm thinking there's a suppression element going on there.
correct. No, I totally
You know,
get I totally get why you would think that, but I'm just saying that I think that his what I was trying, and it could be because I'm not a researcher and I don't use the right terms. Maybe I didn't communicate it with him correct. Because what I was able to get him to say was that we found damage to the mitochondrial DNA. It wasn't like the entire gene was like cut out, right? And I told him and I said, look, that's not what Wallace is saying. Wallace is saying that it just has to be a little hit, and all those proteins encode for the ECT. So therefore, if you hit enough of those, even though they're not completely the protein's not completely destroyed, it's not like complex one is not there, it would be enough to cause the snowball effect. And I think if the mitochondrial damage is not in the mitochondrial DNA, I think it's hard to explain Seyfried's theory, to be frank with you, because if, let's say hypothetically, the mitochondrial DNA was not damaged, well, you would imagine that if the next cell that was created, the daughter of that, it would have a normal mitochondria with normal metabolism, right? So it would have to be that way, I think. And that's where I think that I'm at odds with Seyfried, because I think that otherwise, how does the mitochondrial damage, quote unquote, get passed down to the daughter cells of the cancer cells, right? I don't know how that would make sense unless it was a DNA defect within that subpopulation of cells. I'm just throwing that out there too.
yeah. Well, there's a couple of issues I've got that I would like to just address. One would be, what I struggle with is what's the threshold of apoptosis within a cell when it gets damaged? At what point does that program trigger where the cell realizes it's a detriment to the host? And we've got that whole process is there designed to protect us from
Hmm.
exactly this scenario. So what's being proposed is a certain level of damage is occurring, and as that damage goes down past a certain threshold, apoptosis becomes suppressed, and the cell is able. How is it that that you know how is it that that process of
Yeah.
apoptosis
I think
doesn't
HIF
program itself, it
explains
doesn't
it,
occur?
Mark, because HIF can be stabilized by ROS. So I personally believe that at the beginning, there may not be terminal damage mitochondria, but it's enough to leak enough ROS that doesn't necessarily cause an apoptosis event, but it stabilizes HIF, and HIF then shuts down mitochondrial function, gets rid of the excess mitochondria through excess mitophagy, and that's what kind of forces the cell towards, um, you know, towards the Warburg, type metabolism. But this is me speculating. This is me not
Yeah,
knowing.
Just
is
the
being
other
framed.
contention is as well, it's this notion that if that's the case, so we've now got a cell which has now developed a mind of its own, essentially, isn't it? It's gained autonomy. That's where we're at really with this in terms of, okay, mitochondrial function is lost. Now, mitochondria are regulating reactive oxygen species to communicate and continue surviving because that's the process we're going on here. It's not like mitochondria going, oh, we're damaged, we need to kill ourselves because otherwise they
Hmm.
would initiate apoptosis. So we're basically saying it's got a mind of its own. So why would it produce so much reactive oxygen species? I know it's related to inflammation and damage to the cell, but it seems counterproductive to a cell that wants to survive that it would A, produce that amount of ROS, which is then going to damage the rest of the cell, challenging its own survival. And B, why would, going back to the silver and the honey thing, when honey's supposed to be feeding the cancer cell, why would it suddenly choose to trigger then that apoptosis mechanism, which is allegedly not able to trigger because of HIF1 alpha is upregulated? Yeah, so there's too many contradictions there that suggest that this isn't a process that stops apoptosis because that's just the process. It's contextual. It's back and forth, and there's too many contradictions there that suggest that it's not a cell that is trying to survive by itself because it just doesn't seem to marry with the amount of ROS that's been produced, and then the fact that the cell, even in the presence of honey, glucose, or even silver, decides to then trigger apoptosis.
Yeah, the silver I think is less of a paradox for me because I do think that, metal ions a lot of times generate reactive oxygen species. I mean, that's how ferroptosis happens through iron, right?
Yeah.
Cuproaptosis through copper. Zinc is a toxin. If you do too much of it, it can cause, so I don't think the metal part of it makes as much of a contradiction for me. I think that the honey does, just like fruit, because I do think that most people know, well, in our space, people who are open-minded, we know that honey has a lot of different like enzymes and like polyphenols in it. And I think the question remains, and this is a question I asked to Seafried, asked a question I asked to Durai, why would, if you eat the most polyphenols, which means you're eating the most fruit, why would you have lower chances of dying from cancer? And I personally, and I actually disagree with Thomas, Tomas Durai. I think his thought was, well, the polyphenols are strengthening the rest of the body so you can tolerate chemotherapy better. I don't necessarily agree with that. I personally think that the amount of polyphenols in these foods outweigh the negatives of the extra glucose personally. I think that's my thought. Also, to be honest with you, honey aside, uh, you know, berries are very like low glycemic. They have lots of fiber, like they generally are, they're not like, it's not like you're eating a mango or like a piece of bread, you know what I mean? Like it has a much different glycemic effect, so maybe the fact that it's berries and the polyphenols are helping with the glycemic control and adjusting the microbiome. I mean, there's a dozen, mechanisms you could think of here. But I do think I personally see it as a paradox, and I'm open to thinking that, well, the polyphenols in these foods you're talking about are enough to overcome the negative, that you could have by taking in glucose.
The other thing I'll throw in there as well is the tissue organization field theory. So one of the studies that proponents of that theory did was they took tumor tissue and they placed it in proximity with the healthy tissue, the same type of tissue,
Hmm.
and it reverted back to normal.
Fascinating.
So, I mean, that's why they're suggesting that it's the signaling that's the issue, not the damage to mitochondria or the damage to the DNA, because you had the alleged damage to the mitochondria there, which would be driving the disease.
Hmm.
However, despite that damage, when it's put within that healthy tissue, it reverts back to normal. How is that possible? So from that, I would suggest that that also is a contention against the metabolic theory in the sense that it suggests that mitochondria are not defective to the effect that they are driving the disease. I would argue within that tissue, you've got a much more improved immunological environment which can target the pathogens present within that tissue. The only reason why the tumor forms is because the
Yeah,
pathogen is now protected within that very chronically inflamed environment,
got it.
but you put it back
I
in.
still, I still, I think the only thing I would say to that is that even within your theory, or probably in that theory as well, whenever I mean, if you hear the word Warburg effect, if you
Yeah.
hear the word substrate level phosphorylation or anaerobic glycolysis, that has to equal HIF1 alpha. And if HIF1 alpha is stabilized, the hypoxic response is happening. And one of those responses, and I just, one second, my friend, I got to change my battery in my camera. It just died. Stand
Oh, no problem. Yeah, I thought you were being really still.
by. Yeah, that's not how I look all the time. You know, so. What I was saying before my camera died, if it actually resets here. Okay. So what I was saying before my camera died, basically, was that I do think that a unifying part of what you're talking about, what Navio is saying, and what a lot of mainstream cancer is saying, is that HIF1 is kind of where the crossroads are at. HIF1 sets up the tumor microenvironment. HIF1 programs cancer stem cells. It changes the metabolic reprogramming. Everywhere you see Warburg, you should just think HIF1 is responsible for that because that's what it does.
But
And
what's
so
responsible for triggering HIF1?
that is what's the interesting part, man, because there is, I mean, HIF1 is part of the hypoxic response. It's triggered by hypoxia, right? That's what I was trying to say.
Hmm.
Like, I'm not living at 10,000 feet. I don't have an oxygen, thing. So, like, why would I become hypoxic? Well, there's another area called pseudohypoxia. And
Hmm.
pseudohypoxia is more fundamental at what the triggers are. So, let me just give you some. Circadian dysregulation can trigger it. Inflammation of any source can trigger it. Hyperglycemia and hyperinsulinemia can trigger it. Um, Reactive oxygen species can trigger it. That's kind of the main mechanism of how it gets triggered is reactive oxygen species. But NF kappa B and inflammation, um, certain metabolites of the TCA, citrate, fumarate, succinate, lactate, they can all stabilize HIF. So my point is, even enhanced glutaminolysis somehow stabilizes HIF. My point is there are probably at least 10 that I could find in the literature, causes of pseudohypoxia. And pseudohypoxia causes the same HIF stabilization that it would if you were living at 10,000 feet or if you had obstructive sleep apnea. And I personally believe that's how, and Navio talks about HIF1 alpha in part two of the cell danger response as being the trigger for aerobic glycolysis, right? So I don't think it's at odds. And I think that the chronic inflammation or even the pathogen itself could trigger HIF1 alpha to cause that shift. Is that maybe that's what leads to it? It's just that the problem that I have with that is that at the end of the day, that triggers excess mitophagy, and that's going to get rid of mitochondrial mass. So I still think that they're not necessarily at odds. Both what you're saying and what Seafried's saying is that I personally believe that you offer at least one strong explanation to how the process begins, you know, but it may end up where your theory is the beginning of how the metabolic therapy theory comes into play. And I could be wrong, but I just want to throw that out there.
Yeah, I mean, you could very well be right about that. Um, I think they both can complement each other for sure. Um, and that's the beauty of what I suppose I'm putting out there and what Professor Seafried and colleagues are putting out there as well, is that it trigger more research on both sides into my way of thinking and see if there's some sort of crossover, you know. Um, but yeah, it's always asking the question. I obviously need to focus a little bit more on HIF1 alpha. But for me, I always considered it to be just a response, a pathway which is triggered by something else. So the mitochondria will trigger that pathway in order to elicit glycolysis. So I mean, like you say, you mentioned Crabtree effect. If there's excess glucose present, then HIF1 alpha will be upregulated. But it's just for me, it's just a mechanism for ensuring glycolysis is stimulated in order and insulin is increased, which triggers it as well, in order to get that excess glucose into the cell. Not that HIF1 alpha itself is the driving mechanism, it's just
Mhm. Now, if you
a
look at all
symptom
roads
of.
lead to Rome, dude, if you look at how insulin signals, how mTOR, PI3K, AKT pathways, they all c-MYC, KRAS, almost all of them go through HIF1. HIF1 is the true, I
Yeah.
think, problem, and I think that it really explains a lot of common etiologies, being the the understanding. What I guess I can't, and I don't know the answer to this because I don't know the cell danger theory as well as you do, man. I need to read the papers again and study it, and I don't know, maybe even have that guy on here and talk because I don't know how fast that can be triggered and how much of a stimulus needs to be happening for that to be the case. But it seems like any infection, at least like you're talking about, part of the cell danger response can trigger immediate glycolysis. So it must be pretty quick, you know? And yeah, I think there's I don't know. I don't know everything. I don't want to come off as I know everything. I don't know a lot. I still learn something every day, you know. But I guess, you know, where are we left with, you know what I mean? Like, you know, I guess in terms, and I know you preface this and and you're not a clinician, you're not making any medical claims. I don't want to get you in trouble with anybody, but I guess, you know, talking from like a 50,000 foot view, knowing what you know, um, how does the clinician make a diagnosis of that particular problem? How do you find it, I guess? You know, how do you know what to do about this, knowing what we know? Okay. So I have now watched that segment probably 4 or 5 times. And I just watched it again before I recorded this kind of in segment to finalize this part of our conversation. And I just want to say that I really I really enjoyed the conversation with Mark. I think that we are both pretty convicted in our own kind of belief about how cancer starts and why cancer is how it is and how it acts. I think that there is definitely some common ground. I think that there's things that he needs to learn, and I think there's things that I need to learn about our own respective theories and what they're based off of to probably find further common ground or even tease out the differences a little bit more granularly. I really think that Mark has a lot to offer in terms of his theory. I think that it's extremely interesting. I think there's a lot that needs to be learned. I think he knows that. And I know he's motivated to get the research done to help elucidate the theory, to, God willing, prove the theory, and hopefully allow for diagnostics and therapeutics to be developed so that the theory can be utilized in a way that helps patients in real time. So we will be talking about that in the next segment. If you like videos like this, please like, share, subscribe, and until next time.